First presenter is Dr. Habibur Rahman, and he's speaking on improvising canola using its allied species and exotic germplasm. So, Dr. Rahman.
Thank you very much for introduction, and thanks to the organizing committee, especially Dr. Jennifer Bogdan, for inviting me to make this presentation. So I'll be talking about improving canola or breeding canola by using exotic species, allied species and exotic germplasm. So as the audience of this attending this conference is from very diverse backgrounds, so therefore I'll be giving a very high-level presentation, not going in depth of much. So here in this slide, I am showing some of the important events related to plant breeding. So since the discovery of Mendelian genetics, plant breeding is very much focused on scientific principles, but events related to plant breeding can be traced back 1760, where hybridization has been performed for the first time. So now the question, when plant breeding has started, is it about 250 years ago or longer? So based on different evidence, we believe that agriculture has started about 10,000 years ago. At that time, humans started to domesticate the crop plants and use for their own use for food and non-food purposes. So at that time, what they have done, they have domesticated the wild crops for their own for growing in their farm or somewhere. So on the right side top, I'm showing this wild type and domesticated type of different crops. And you see that's compared to this wild type, domesticated type has better phenotype, that is appearance. So how this better phenotype appeared, this is due to mutation. And mutation, what is mutation? Mutation is change in the gene, okay? So when the ancient people, they were selecting the crops for their use, they must have selected the improved one. That is, they have made some genetic improvement without any scientific knowledge. So based on this, we can say that plant breeding has started about 10,000 years ago. On the right side, right side here, I'm showing this sphericals, so with a different color, they represent different genes and alleles. You see, compared to this modern cultivar, there has been wide diversity in wild progenitor, and then diversity reduced due to domestication, and then modern breeding has further reduced diversity. So what happened? That's in plant breeding, we always select some desirable genes and allele and discard the undesirable one. But while we are doing this, sometimes we lost some of these desired allele. Therefore, sometimes in a crop species, we may not find a trait, but we can find the trait in wild germplasm. So here I'm showing an overview of the use of wild relatives in crop breeding of different crops. You see there's a lot has been used to improve disease resistance in crops, and also the wild species and relative wild species and crop relatives has been used for improving abiotic stress tolerance, yield, and quality traits also. Turning to canola, it's an important crop in Canada. So you see that over the year, yield, how much has increased. So this is European, and then here blue one is this Canadian canola. Since launch of hybrid in early 2000, you see that there has been a little stiffer increase in yield compared to earlier years. But if we look at maize, you see that's hybrid was launched in 1940s, and then you see that's how much stiffer yield increase has occurred, and that has been achieved through intensive research and breeding. So what is important thing for hybrid breeding, we need to increase the level of heterosis. What is heterosis? Heterosis is this excess performance of the hybrid compared to their two parents. And to achieve this, or to get this high level of heterosis, what is important? Diversity between these two parental lines. So here I am showing a very well-known triangle of Brassica triangle. In this triangle, I will not go in detail all of this. So you see that's the Brassica napus canola here, and it evolved from interspecific hybridization between Brassica rapa and Brassica oleracea. Brassica rapa carry the A genome, and oleracea carry the C genome. Molecular analysis of our fingerprinting, genetic fingerprinting, has identified this Brassica napus is very distinct from Brassica oleracea and Brassica rapa. So therefore, this wide diversity of Brassica oleracea and Brassica rapa can be used in breeding to improve Brassica napus canola. Also, within Brassica napus canola, you see the spring type, which is grown in Canada, and then winter type and rutabaga, they are genetically very distinct. So therefore, this type can also be used to improve Canadian canola. So I'll be presenting some of the results from our research on the use of these types. First, using winter canola and rutabaga. So we crossed several accessions of winter canola and rutabaga with spring canola and applied different types of breeding methods, double haploid, pedigree, and different breeding methods, and analyzed the population with molecular markers, and then develop a lot of genetically diverse line. And from this, we are able to develop some open pollinated cultivar, for example, UA alpha gold, which was giving about 12% greater yield than the open pollinated check at that time, and then we also developed some other cultivar. So what is needed in this type of cross? Intensive selection against vernalization. Vernalization, what is this? This winter canola and rutabaga, they will not flower without exposure to low temperature. So we had to make intensive selection against this vernalization gene, as well as for earliness of flowering and maturity gene. And here you see that so we selected this plants, spring type plants in greenhouse, and then when we took to them in the field, and then you see some of them were not flowering at all. So repeated cycle of selection was needed, and then we achieved this spring type canola. And then also during this time, intensive selection for canola quality traits were needed. Here I am showing the performance of different types of line from our breeding program. So lines from spring by spring crosses, which is normally canola breeders do, and then winter by spring crosses, rutabaga by spring crosses, and then gene bank, which are open pollinated old cultivars, and some industry cultivars. So you can see among these, this winter by spring cross lines, they are giving the greatest yield, mean yield, and also large variation, some line with more than 20% seed yield. Spring by spring and rutabaga by spring, they are giving similar performance, but I will show in the next slide, this rutabaga by spring material, they were giving greater level of heterosis, which is needed for greater yield in hybrid. And gene bank material and then industry lines, they were much poorer. So here I am showing some comparison of this inbred line derived from spring by spring cross and then winter by spring cross. And there, the hybrids of spring by spring cross line and hybrids of winter by spring cross line. So you see that's compared to this spring by spring cross line, winter by spring cross line yielded much greater yield, and that hybrid also gave much greater yield compared to these hybrids. So it seems that, yes, winter canola germplasm can contribute genes and allele, which can increase yield of open pollinated cultivar as well as hybrid cultivar. So we also estimated the mid-parent heterosis, which is an indicator of that how much genetic or what type of gene contributing to this heterosis. If it is a mid-parent heterosis is high, so that means that non-additive type of gene is contributing to heterosis. So here you see this compared to the spring by spring, winter by spring, it gives almost the same level of this mid-parent heterosis. So that means that winter by spring cross material or winter parent was not contributing that much non-additive type of gene effect for increasing the level of heterosis. They contributed additive type of gene, which increased the seed yield of hybrid, but not much non-additive type of gene. So here I am showing this yield performance of this rutabaga inbred line, and then their hybrids. You see this inbred line of rutabaga cross, they were not better than the Czech spring canola Czechs, but their hybrids was giving some of these hybrids was giving greater yield. When we make correlation between the seed yield of the inbred line and seed yield of the hybrid, there was no correlation. So this indicates that non-additive type of gene effect was contributing to a higher yield in the hybrids. So we also fingerprinted this inbred line derived from rutabaga cross, and then estimated their genetic distance from the spring canola parent, which has been used for production of hybrid. So genetic distance was showing positive correlation with hybrid yield, positive correlation with mid-parent heterosis, and then also mid-parent heterosis was contributing or giving a positive correlation with the hybrid yield. So all these evidences support that non-additive type of gene effect was contributed by rutabaga, and therefore we are getting higher yield in hybrid. We also used winter canola and rutabaga for integration of clubroot resistance, and then we applied similar types of breeding method, what we have explained, what I have explained in the earlier slides. In this case, we had to make selection for resistance to clubroot disease, and through this we developed a lot of canola line carrying clubroot resistance, and then we mapped this resistance, and then I am not going in detail of mapping. So we identified a locus on chromosome H3, and based on marker of this chromosome, we constructed the physical map of this chromosome using 2 reference genome. And then by comparing this genetic map, the position of the locus, and then physical map, we could deduce that this locus is located at around 20 to 25 million base pair of chromosome H3. And then we also developed marker, and then showing co-segregating with the resistance. And then you see this marker for resistant, marker for susceptible, this marker for resistant, susceptible. So we used also Brookfield and polycross rutabaga for integration of resistance as well as for mapping of the resistance. And then we developed several canola line carrying rutabaga resistance, and from there we developed a non-GM open pollinator cultivar, because I heard there was a demand for non-GM canola. And then also we supplied this resistant line carrying rutabaga and resistant line carrying Mendel resistance to the industry to develop clubroot resistant hybrid carrying both resistance A3 and A8 resistance. Some of this cultivar has been marketed by industry. So we mapped this resistance, clubroot resistance of Brookfield, and then we identified 2 loci, one on chromosome one on A8, which is giving a major effect to the resistance, and this give also resistance to multiple pathotypes. And then in addition to this, we identified a locus on chromosome A3, and which is giving very small effect to the resistance. And chromosome A3 locus is located around 14 to 15 million base pair position. So remember that this locus was giving a major effect, and this was giving a minor effect. So we also mapped the resistance of polycross rutabaga, and in this case, we have done a little more in-depth study. So we sequenced this resistant and susceptible line, and based on the sequence, we identified the SNPs, single nucleotide polymorphism, and then based on SNPs, we developed marker and then mapped this resistance on 2 chromosomes. One is A3 and another is A8, same. Okay, but in case of A3, so here is the same, but it's giving a major effect. On the earlier case, it was giving minor effect. So what indicates, even in rutabaga, different form or different alleles can be found for the same resistance gene. So this A3 is located around 14 to 15 million base pair, and then A2 is 10 to 12 million base pair. Another interesting thing with this, that these two loci, A3 and A8, in addition to direct effect, they also contribute to the resistance through interaction of these two loci, which has not been much reported. And this is, I repeat, this is the major locus, and this is the major locus, and their interaction contributing to resistance. To my knowledge, this is to date, is the best source of resistance. And then we developed from the SNPs, SNP allele-specific marker, and then you see this marker is getting with resistance, and susceptible are lacking this marker. So we went a little more in depth of this, try to identify which gene is contributing to this resistance. For this, we took 30 genes from this genomic region and from these two genomic regions, around 15 from here, around 15 from here, and carried out expression analysis on the line carrying either A3 resistance, which is blue, and or A8 resistance, which are red, and then no resistance susceptible. And then we found that from A3 gene, to sugar transporter gene of A3 was significantly or showed significant reduced expression in the line carrying A3. In case of A8 gene, 3 TNL genes, which showed significantly greater expression in the line carrying A8 resistance. So based on this evidence, so we can say that yes, sugar transporter gene of A3 and then TNL gene of A8 might be contributing to this resistance. So we also crossed Brassica napus canola with Brassica rapa to develop genetically diverse germplasm. I will not be going in detail of this interspecific cross because a lot of researcher has been doing or using this interspecific cross for developing canola, improved canola germplasm, as well as for improving earliness of canola, and then also integration of clubroot resistance. So we also integrate clubroot resistance from turnip and Chinese cabbage, and then map this resistance, and then this resistance is located on A3, and then we develop marker for use in breeding. And then we developed a lot of genetically diverse line from this interspecific cross. So one thing is very interesting and academic, and you see that say here, this when we make this interspecific cross, the F1 is AAC, that is a genome has its homologous chromosome, pair chromosome, but C genome has only single set of chromosome. Theoretically, what was expected, during cell pollination, this C genome chromosome will be sorted out or eliminated. That is, in the progeny, we will have only AA, that is Brassica rapa type. But in practice, all stabilized into Brassica napus type, which is an advantage for breeders to use this interspecific cross. So now, next slide, I will be presenting some of the results of use of Brassica oleracea in the breeding of canola. This interspecific cross has not been attempted by researcher or breeder because it is extremely difficult to make this interspecific cross and get a F1 hybrid plant. For this application of in vitro cell and tissue culture technique, for example, ovule culture, embryo culture is needed. Even we culture this, it's very difficult to get a hybrid. So we are able to, we put a lot of effort to develop interspecific hybrid of this crosses. And when we get the hybrid, and then you see that in some cases, you can get a plant like this. You will not like this at all. And then also, you see that seed set, almost no seed set. We had to manually self-pollinate thousands of plants to get the progeny. How about through intensive selection and breeding, we are able to develop canola quality line from this cross. And like this napus rapa cross, here also F1I hybrid was like this genomic composition, but it's stabilized into Brassica napus type, not into oleracea type. So I will be giving some example of integration of alleles from oleracea for improving canola. So on the left side, it's not cotton field. Don't smile, it's cotton field. It's canola field. It's before harvest. So you see this earliness of flowering and maturity is important for Canadian canola. And on the right side, I'm showing that you see that's this triangle of U, all these 6 Brassica species. Brassica napus is here, and Brassica rapa. You see that it has finished flowering almost, and Brassica napus started to flower, and Brassica oleracea, the other parent of Brassica napus, has not yet flowered, or there is no sign. So based on this, one will assume that, yes, earliness of Brassica rapa can be used to improve canola. Yes, that's right. That's a lot of breeders, they've used this. But in oleracea, even it's very late, but there's some genetic variation or phenotypic variation for flowering time. So we thought that the early flowering Brassica oleracea might carry flowering allele, which can improve earliness in Brassica napus. To prove this, so we made interspecific cross of canola and then Chinese scale, and then developed an F2 population, and then F2 population showed wide variation in flowering, and we selected this most early flowering one and grown next generation and continued selection, repeated selection for earliness of flowering, and after that we achieved an F7 line, F6 and F7 line, which was flowering, you see, much earlier than this Brassica napus parent, canola parent. And it was very close to Brassica rapa. Then we mapped this to further. This indicates that when it's flowering much earlier, then we crossed these two species, and then we got something, this. Then it must be this early flowering allele derived from Brassica oleracea Chinese scale. To further confirm that, we mapped this resistance gene, mapped this flowering gene, and then this flowering gene is located on C1 chromosome, and this one of the unique characteristics of this locus is that it was not influenced by environment, as well as it was not affected by photoperiod. And this has an advantage because when it's photoinsensitive, this locus or allele can be used to improve Canadian canola, as well as that can be used in other parts of the world where canola grown under short condition. So we also identified an allele of Brassica oleracea contributing to high oil content, and this was on C5 chromosome. So in this case, the allele from Brassica oleracea was increasing about, this is the napus allele effect, oleracea allele effect, and then it was giving about 1% increase in oil. 1% probably you will think it's a very small amount of increase, but considering this canola contains about 45% oil, and then if you make a math, you'll see that per year, knowledge-based use of this allele will contribute several million to the Canadian economy. So we used also a lot of different types of vegetable brassicas. So that's cabbage, cauliflower, broccoli, Brussels sprout, and a lot. And we applied the similar breeding method, and then we developed a lot of canola line. And then we tested around 228 canola line, and then mapped different loci, controlling traits. And then you see this, we identified 2 loci for flowering time on C2 and C5. And in this case, C5, the allele of cabbage allele, so it reduced flowering. So that means that early flowering allele can be found also in cabbage. We also identified locus for grain filling period, and then oil content and glucosinolates. That's in the next slide. I will be summarizing some of the results here. You see that by using this exotic germplasm, we were able to disclose some of the loci which has not been reported previously. Because for identification of a locus, we need contrasting allele. If in canola we don't have contrasting allele, we will not be able to disclose this allele or locus. So by using this diversified germplasm, so we were able to disclose new locus. You see that's a QTL for GLS glucosinolate oil content and the seed oil content and grain filling period and glucosinolate, seed oil content. And we also identified many locus, you see that's which has been reported by previously by different researchers. So another advantage of using this exotic germplasm that we can identify unhidden or undisclosed loci for use in breeding. So we studied this impact or this value of this germplasm diversified with Brassica oleracea for use in hybrid breeding. So what we have done, this I showed this line in the earlier slide. This line we crossed with canola and then produced test hybrid. And then tested this hybrid in field trials. We tested in 4 field trials. And then I'm showing here, this is red is inbred line, and then blue is test hybrid. So you see this performance of the inbred and test hybrid overlapped for all traits, except for seed yield. In case of seed yield, you see this performance of the inbred was lower compared to the performance of the hybrid. So this indicates that this alleles from Brassica oleracea can be used to increase seed yield. And how it can be increased? And then here I am showing a comparative view of this of the line derived from spring by spring cross, and then spring by Brassica oleracea cross, and then their line yield, this inbred line yield, and then inbred line yield of this, and then their hybrid yield, hybrid yield. You see this compared to this spring by spring cross line, and this oleracea cross line, they gave much lower yield as inbred line. But when we look at their hybrid yield, and then here, you see that this is giving much greater hybrid yield. So we estimated the mid-parent heterosis. What is this mid-parent heterosis? If you remember, there's this types of amount of non-additive effect genes. So you see this compared to spring by spring oleracea cross was giving almost double magnitude of or mid-parent heterosis, that is non-additive effect type of gene. So that means that Brassica oleracea was contributing a lot non-additive type of gene effect or non-additive effect alleles in canola to increase the level of heterosis and thus to increase the yield of canola, hybrid canola. So we also introduced clubroot resistance from Brassica oleracea kale, and then for this we crossed and developed F1 and self-pollinated F1 for several generations with selection for clubroot resistance, and then we also crossed this with canola, and then we self-pollinated this backcross population, but we are not able to get any clubroot resistant plant from this stream. But from this stream, we are able to get a clubroot resistant line. And then for this, we have to test more than 11,000 plants and then self-pollinate manually. There's a number of plants to get some resistant one. This conclusion, favorable allele for agronomic and seed quality traits can be found in exotic germplasm allied Brassica species for use in knowledge-based canola breeding. What does it mean, knowledge-based canola breeding? I showed this one allele was giving about 1% increase. So we have to develop marker, allele-specific marker, and use in breeding. That's the knowledge-based breeding. And exotic alleles of winter canola increases seed yield in open-pollinated and hybrid spring canola. And the alleles of rutabaga and oleracea can increase the level of heterosis for seed yield in hybrid canola through non-additive type of gene effect. Clubroot disease resistance of winter canola, rutabaga, rapa, and oleracea can be used in spring canola breeding, and allelic variation can exist for clubroot resistance loci, and non-allelic interaction between loci can play an important role in clubroot resistance, which I have shown by using this polycross resistance. With this, I would like to thank all this funding agency for funding my research, and thank you for your attention.
Yeah, I think we have time for maybe one quick question if there is one. I guess not. Okay.
Oh, yes. Yes.
Oh, now we've
Yeah.
got two.
Oh, sure.
I was just curious on the last slide on the cave plants using Brassica oleracea plants. I just
Okay. Why this backcrossing of F1 to canola, and then from this backcross one derived population, why we are not able to get clubroot resistance? It's due to that could be a population size, smaller population we used here, and then also the type of gene effect. It's quantitative trait in Brassica oleracea, clubroot resistance, and then probably we're not able to capture all this. And then also there might be some recessive type of gene effect contributing to clubroot resistance, and that might be also contributing to this. There's one question.
Yeah, I think we had to move on to keep things
Okay.
online on time.
So I'll be there. Yeah.